Good morning. It is now my pleasure to introduce our next participant, the CEO and founder of SQZ, Dr. Armon Share. The format for the session is approximately a 25-minute fireside chat, and if you have any questions, feel free to enter them into the chat box underneath the Zoom window, and we can try to ask them on your behalf. So, Armon, thanks for joining us today. Maybe we can start with an overview of SQZ's unique platform, including the APCs, AACs, and TACs, what all of that means, and more generally, your approach to cell therapy.
All right, thanks a lot, Gula, and thanks to you and the Chardan team for having us on. Always good to chat. And yes, I know we have a lot of acronyms, so it makes it complicated to follow sometimes. But just for the background on SQZ, so the company is founded around this basic discovery that if you take cells and squish them at high speeds, hence the name, you can actually disrupt their membrane temporarily and allow anything that's outside to diffuse into the cell. And then the cells just detect these disruptions and close it back up. Now, it sounds like, a very simple discovery because it is, but it solves a very fundamental problem when it comes to how do you think about creating cell therapies. Because right now, I think we're just at the beginning of what cell therapies can do in the long run. And it is currently very difficult to actually engineer the interesting functions you want within a cell. And it's very hard to scale that such that it could be kind of time and cost effective from a patient perspective and from the perspective of having an ultimately broad therapeutic. And so with SQZ, by solving this issue of just how do you put stuff into cells, it seemed like we could open up a lot of doors as far as what cell types one could now engineer, what kinds of material classes could go into them. And finally, it appeared we would otherwise keep the cells normal. And so these three factors meant that we could now engineer all kinds of things about cells that people would have had a lot of trouble with in the past, because the main techniques right now are viral transduction and electroporation. And these techniques tend to have a lot of constraints on cell type, cargo, and they often cause cell dysfunction. So for us, it opened up a lot of biology that we could now do that was previously very difficult for people. And the second component is that because it's such a simple concept where it's just microfluidic membrane disruption, it meant that from an engineering perspective, this has been much simpler to implement. So our current trials have all been under 24 hours to manufacture the cell product, and vein-to-vein time is about a week for our patients. So it's a very different feel from what people have experienced so far. And we are moving towards a point of care version of the system, meaning that we've integrated the SQZ unit op with all the other unit operations that do the washing, et cetera, such that it's all one system that has disposable kits. So now you wouldn't need a clean room down the line, and you can actually automate and simplify the operation such that someone in the hospital could do this. And that could really further improve the accessibility of these cell therapies. Now, to give a sense for kind of on the biology side, what we do and what all those acronyms are, we ended up going a bit of a different route than what most cell therapies have been doing, which is we tried to go for those areas of biology that were very hard to do for existing techniques, but we felt SQZ was going to be very good at opening up new spaces. And one of the big areas there has been to go into the kind of controversial zone of cancer vaccines, because cancer vaccines are one of those things that they've always been a great concept if you could get it to work, but no one's gotten it to work. And the underlying principle for a cancer vaccine is, can you get a good CD8 T cell response against the tumor? Because there's plenty of evidence in IO and other cell therapy work that CD8 T cells are great killers when it comes to the tumor, and they could be an excellent driver of efficacy in solid tumor settings. But it's been very hard for people to get a good CD8 T cell response going. And one of the kind of immunological cores of that problem is that right now, if you think about the vaccine modalities that people use, be it for infectious disease or in cancer, they all ultimately rely on a process called cross-presentation. And what that means is you have antigen that you want to activate a response against, and that antigen, no matter whether you were an mRNA vaccine or a viral system or what have you, that antigen ultimately is getting endocytosed by an antigen-presenting cell. And when antigen gets endocytosed, it primarily ends up on MHC class 2, which gives you CD4 responses and antibody responses. Very little gets cross-presented onto class 1, which is what you need for CD8 responses. And with SQZ, we've been able to show that we can just load the antigen directly into the cytoplasm of these APCs, and then that's how we can get much better class 1 presentation and drive a strong CD8 T cell response. And we actually had a paper come out in Journal of Immunology earlier this year that kind of gets into the details of that. And so 2 of our clinical programs, what we call SQZ APCs and SQZ enhanced APCs, are based on this principle of we can load antigen cytosolically and now get much better class 1 presentation to drive a CD8 T cell response. And the enhanced APCs take that principle and add certain functions on top of it by having things like membrane-bound IL-2 and 12 and CD86 to collectively drive the T cell response even harder. So both of these programs have readouts coming in the second half of the year. The APC program, which is more advanced as it is the first generation of it, already had some interesting data at ESMO IO where we saw a nice clinical response in our high dose cohort where all the biomarkers also really added up, showing massive CD8 infiltration into the tumor. So, happy to go more into any of those zones. And then kind of to close it out on what other programs we've been doing, we have the AACs, which is our red blood cell based mechanism. And this is taking a bit of a different principle where we're actually using red blood cells as kind of a Trojan horse to deliver material to professional APCs in vivo. So what we do is we take a patient's blood, squeeze in antigen plus adjuvant, and make these red blood cells look aged. And aged red blood cells, when you inject them, primarily get taken up by professional APCs. So now you basically have a carrier that's going to very effectively drive a lot of antigen and adjuvant into these professional APCs and make them activate the immune response for you. So that program also is going to have a readout in the second half of the year. And beyond that, we think SQZ can be applied to many different areas. So the next most advanced work is our work in autoimmune diseases, where actually we also had a paper recently in Frontiers of Immunology that describes that system. But basically that also leverages red blood cell based mechanisms to shut down immune responses in an antigen specific way. And we're pursuing it for celiac disease first, but we've already shown that it could be applied to things like type 1 diabetes as well. So that's probably the quick-ish overview of what we've been up to, but happy to dive into any areas you'd like.
Yeah. So you touched on a couple of points there that I want to expand on a little bit. So you mentioned some ways in which this is different from how people typically think of cell therapies. People do tend to lump cell therapies together with respect to perceived complexity. So maybe can you discuss a little bit more the advantages of your technology and what sets it apart from cell therapy platforms that people might have in their minds?
Sure. So I think an area that people don't often think about because of the constraints that exist is how limited how limited the field is today on what kinds of biology can be engineered in a cell. Because viruses, for example, only work for certain cell types, and really the only thing they can do is insert a gene. They can't delete something. They can't just temporarily upregulate or downregulate. It's a pretty coarse tool to just insert something. And by the way, they can cause all kinds of trouble. And then the other alternative is things like electroporation, where you're basically zapping the cells in an electric field. And that can work for, again, certain cell types and certain charged materials, but it's otherwise got restrictions and can also be limited on the material classes it can work with. So with SQZ, because it really opens up that, this has worked for every mammalian cell type we've tried, it works across material classes regardless of charge or chemical composition, et cetera. You can now go after a lot of biologies that are different. And a lot of what people associate with cell therapies right now, like the need for preconditioning, or the fact that because you're causing a genetic change, you're going to have long-term safety concerns for how this works, or things like CAR Ts are heavily associated with CRS, et cetera. Cell therapies have a certain profile of being a potentially impactful, but otherwise costly, toxic therapy that tends to be at last line. And with SQZ, by releasing that kind of biological constraint, it means you can go after biologies that could be very relevant for early lines. Or by not touching the genome of these cells, you can have a much cleaner long-term safety profile because you don't need to worry about, did I cause a malignancy by touching the genome of the patient's cells. So it lets you go after other biologies like the cancer vaccine biology I mentioned, or the autoimmune biology I talked about, where these are all things that could be very relevant to early line because they have much better toxicity profiles and are going after more physiological mechanisms that you would want in those early settings. And kind of the final piece is that time and cost issue, where because from an engineering perspective, Squeeze is so simple, and you have all this biological flexibility, you can have processes that are much shorter. So none of our processes involve cell expansion, and they obviously don't involve viral transduction. So that dramatically cuts the time. And a lot of cost for cell therapy is actually primarily correlated to manufacturing time, because your cost of the clean room and the operators is one of your biggest drivers of cost. So if you take cell therapies that normally take weeks, but instead you have a cell therapy that's under 24 hours, you're already saving an order of magnitude in cost because you've cut down the time. And the final piece of that is because of the engineering simplicity lending itself to being eventually a machine that could be at the point of care, now you're also transforming the logistical dynamics around cell therapies, where you don't have to assume you're going to send everything to a central site, deal with all the logistics of getting it back. Did I track the right patient? God forbid, did I put patient 1 material into patient 2, et cetera? By being able to do it at the hospital site, you can simplify a lot of that and make these things much more accessible and scalable.
To expand on some of the things that you just mentioned, particularly with regard to, let's say, point of care and release testing, and how do those things fit together? So, you talked about, short process overall in terms of the manufacturing time. What is the FDA has been focused in recent times on things like potency assay release testing regarding cell therapies. So with your platform, how did those issues play out in terms of what you think regulators would want to see with the current iteration of your or incarnation, I should say, of your technology, and as well as with the point of care potentially?
So I think one important kind of baseline element of this is, because for all of our therapeutics, we don't do any genetic changes, that already dramatically simplifies and releases a lot of the constraints that the FDA would otherwise be concerned about, because we're not risking any kind of long-term tox issues. And the unfortunate thing with the risks of genetic changes is it literally takes 1 out of 1 million events to be enough to cause trouble for you. So there's a lot you would want to verify, especially as you get into a therapy that's going to be more broadly available, or something that's going to be used for earlier lines, et cetera. So for us, because all the alterations are transient, that automatically takes away a lot of the safety concerns and release elements that people would otherwise think about. And what we have right now from our interactions is really oriented around viability of the cells, identity, and some level of characterization for potency or content. And at least for things like sterility, viability, identity, et cetera, these are all things that, as you think about an integrated point of care system, you might be able to start to take away some of these tests. So, for example, with a point of care system, if it's a closed system, which just has sterile disposable kits, we think there's a pretty strong case to be made to remove the need for sterility testing. Our sterility testing right now is already a rapid sterility test, so it's about 2 to 3 days. So it's already much faster than the compendial 2-week approach. But we think you might even be able to get rid of that 2-3 day test if you have a closed system where it's patient material on, sterile system, patient material off, and you're just disposing of every contact surface with the cells. So these are things that we think can simplify it, and then your release can hopefully just be flow-based things or other things that are relatively practical to do in an on-site setting. Now, for the potency assay side of things, I think for situations that I think that most comes to mind is things like TILs, where it's been a whole thing about what's the potency assay going to be, how much trouble is that going to be. I think that's less complicated when you have clear elements of the cell that you've engineered that you can point to, versus if you just took something from the patient, expanded it, and want to put it back in. It's a little harder to clarify what potency means. And so that's where for us, we've been trying to make sure we have those discussions early on. And already there seems to have been a trend with the FDA where they care to hear about potency assays earlier, whereas they didn't used to necessarily care as much. So we get to have those conversations earlier with them. And for all of our programs, we think we have a pretty good handle on it because generally there's something that you can point to that you actually engineered about the cell that can reflect the potency of it. So for example, with our APC program, we have T cell clones that can respond to any presented antigen on HLA-A2, MHC class 1. So that potency assay is pretty clear. For things like the enhanced APCs, where we have other domains like membrane-bound IL-2 and 12 and CD86 on the surface, you can directly measure those or some derivative function of them, and that can serve as your potency. So there's more flexibility in that way on how you show it.
Got it. That makes sense. And so you talked earlier about the different cell types that your platform can be used to modify. So it seems like it can work for either autologous or allogeneic cell therapy. So can you discuss what you've learned looking at both of these avenues, for example, with red blood cells and peripheral blood mononuclear cells, and what you've learned regarding manufacturing considerations?
Yeah, and that's definitely been an interesting, almost counterintuitive elements. So, because Squeeze inherently is agnostic to doing something autologous or allogeneic. I think on the biological side, the thing that one needs to keep in mind when considering allo versus auto is that, let's go for the category of immunologically related phenomena. When you go allogeneic, you're going to have to deal with some level of graft versus host problems because whatever that allo source is going to recognize the patient's body as different. And so you're going to have some level of graft versus host issues. And in parallel, you're going to have rejection issues because the body is also going to recognize the allo cells as foreign. So you're actually adding biological complexity when you do this. And often that means you're going to be sacrificing safety, efficacy, or both. As a result of going allogeneic. So that's where for some things we think autologous will always be biologically more favorable. And if manufacturing and cost are a very different dynamic with a squeeze-based system, why bother going allogeneic? So that's kind of one dimension of it. And even for some situations like red blood cells that you alluded to, it's kind of non-obvious what these issues are, but when you dig into it, you see it. So we have two sets of RBC-based therapies. One is the AACs on the oncology side, and then TACs for the tolerance side. And we frankly initially assumed we would go allogeneic with those. But once we dug into it, we saw that it's not actually that simple because you think O-negative blood is fine to inject into anyone. But actually, there's a lot of minor antigens you need to be aware of. And minor antigen mismatches can lead to problems if you try to repeat dose anything. So that's where we actually started to find that it was getting more complicated to do an allo therapy, and there were more things you would have had to verify from a release testing perspective, et cetera, whereas an autologous therapy would have been much simpler. And I'm not saying this is necessarily a biological argument, but as you think about non-life-threatening indications, patients can tend to view things more favorably if you're saying, I'm taking your blood, engineering it and giving it back to you versus I'm taking someone else's blood and injecting you with it. So these kinds of little factors on the patient side can come to light too. So, long way of going to, in the end, we actually found if you're not constrained by the time and cost issues that typical autologous cell therapies are, a lot of times you're actually much better off going allogeneic because you would expect better efficacy and safety depending on your mechanism. And you can actually reduce the release testing complexity or other manufacturing complexity because there's far fewer things to be concerned about and test for when you're doing something autologous.
That's really interesting. Okay. And so regarding your manufacturing capabilities, you talked about how the process works, the different cell types that it could be applied in. Let's talk about scale and current capabilities and how you see that evolving as you advance through your clinical programs and move towards commercial potentially.
Sure. So from a scale perspective, the basic principle of how squeeze works is you basically have a microfluidic channel, and then there's a constriction element, and the cells go through that constriction at high speeds. That's what disrupts the membrane and allows material to go in. So the way we've increased the scale from research to manufacturing is you basically just have microfluidic chips that have more channels in parallel. So the research scale is on the order of tens of channels in parallel. The clinical scale is almost 1,000 channels in parallel. And this chip is like a third the size of your business card. And so that chip does 10 billion cells a minute for the clinical scale. And a typical leukopak, for reference, is on the order of 10 to 15 billion cells. So the squeeze step is actually the fastest manufacturing step there is in our production process. And the rest of the chain is relatively simple unit ops because it's just things like washing and buffer exchanges or putting them in cryomedia and freezing them, et cetera. There's no expansion. There's some centrifugation stuff for certain separation. So it's all in all a pretty simple system. And the way that's implemented right now is that we have a process chain in a clean room, and you can just replicate that process chain if you needed to expand capacity. But an important thing to keep in mind is because our manufacturing time is under 24 hours. So for our lead trial, the APCs, it averages around 17 hours. You can get a lot of patients through the same footprint pretty quickly with this. So what I mean is, if your manufacturing process is under 24 hours, let's say you have a clean room that's running 24/7, within a month, you can probably do 20 or 30 patients. Whereas if you had a manufacturing process that was, let's say, 2 weeks, you could do 2 patients. Or maybe you parallelize a little bit, you could do maybe 3 or 4 in that same footprint. So it dramatically improves your efficiency of the footprint, and it's easy to replicate what it is. So we view this as kind of really a scale out model, but you get a lot of mileage per kind of unit area or per process line. And that's with the current central manufacturing setup. For the point of care version of the system, that's a totally different game. It's an integrated machine, disposable kits. And for that one, you just up the number of machines to up your capacity. It's a pretty straightforward system.
Great. And so we'll talk more about this in the upcoming cell therapy panel, but to what extent can your capacities or capabilities bring cell therapies into mainstream clinical care?
So that's definitely our aspiration, is to really make cell therapies that are going to have a dramatic patient impact, but also make them much more accessible. And I think if you can make a cell therapy be much simpler for a patient from a perspective of, I'm not waiting months to get this, and from a payer perspective, this isn't going to be cost prohibitive to do, you can really transform that. And that's where I think things like a point of care system are key, because now you're dramatically simplifying what it means from a time and cost perspective for these patients, but you're also really improving the scalability and accessibility of this. Because if you have an integrated machine that just needs a trained operator or two, most hospitals should be able to handle that. And now you can have therapies that could be applicable to millions of patients instead of a few thousand, because it's a system that can be broadly implemented. It doesn't need to be the very specialized academic hospitals.
Great. We'll definitely be talking more about that in the upcoming cell therapy panel today, so everyone stay tuned. And I think we're at the end of our session for now. And so, yeah, thanks everyone for participating and for the discussion, and we'll check back in with you soon for the panel.
All right, see you in a bit, Gula. Thanks a lot. Bye.